All right, let's go ahead and start talking about our lecture today. Before we head into photosynthesis, though, I know there are a couple things that are due today that you may want to hand to me in person. I'm going to say at the end of lecture, we'll just have a pile. Maybe we'll make a pile at the end of this table that you can hand it in, instead of taking the couple minutes to shuffle papers around here. That being said, today's lecture is pretty content heavy, all right? I understand it, and I understand as we go through it this first time, you might not understand it all completely, and this is probably going to be content that you're going to have to review, all right? I have tried to simplify it as much as I possibly can, so please ask me questions if you are stuck. So we're going to look at photosynthesis today, all right? And photosynthesis is kind of similar to cellular respiration. It's kind of the opposite, right? So if we look at this cycling of energy here, we see in cellular respiration, we are using organic molecules, often glucose, with some oxygen to create ATP. And as some byproducts, we get water and CO2, right? Now in photosynthesis, we're going to use those byproducts of cellular respiration, CO2 and H2O, as well as some light, right, to create organic molecules, primarily glucose. Right, so it's this nice cycle. Right, and some of these steps are going to be similar to steps that you have seen in cellular respiration. All right, so overview of photosynthesis. Right, we are using carbon dioxide, we are using water to create a sugar, primarily glucose, and we get some oxygen as a byproduct. This reaction as a whole is not energetically favorable, right? If we look at this Gibbs free energy here, it's a positive value and it's a whopping positive 685, right? That is not energetically favorable. This reaction is not going to happen spontaneously. We need a big source of energy for this reaction to happen. And in this reaction, we get that energy from sunlight. Right? Light from the sun is going to provide the energy to make this reaction exergonic and actually go through these processes. All right. So if we're talking about photosynthesis, right, we're talking about organisms that photosynthesize, right? And if we took all organisms on Earth and broadly split it into two categories, we would have heterotrophs and we would have autotrophs, right? Heterotrophs need to eat food in order to get energy, right? We're autotrophs. The pets you have at home are autotrophs. Anything that needs to consume food. Yes. Yes, they are heterotrophs, yes. Thank you for correcting me. I was saying autotrophs because I'm thinking photosynthesis. Yes, if you eat food, you're a heterotroph. Thank you. That was my slip up there. All right, so we're heterotrophs. Your pets are heterotrophs, right? Unless they're some strange mutant and they have chloroplasts in their cells and then can photosynthesize. All right, so then if we're talking about those autotrophs, all right, these are organisms that don't need to consume food. They get and make their own food from inorganic sources, all right? So we can have autotrophs that get food from like a type of chemical energy. We call these chemiautotrophs. Often we see these in like deep sea organisms. But the organisms we're going to focus on today are photoautotrophs. Right? They are getting their energy using light as their energy source. Right? We often think of these as being land plants, but there are a whole host of other species out there that can photosynthesize that are not plants like grasses and trees. Algae, cyanobacteria, little tiny organisms here. Some of the protists, like Euglena, right? They have chloroplasts. They can also photosynthesize. We're going to focus on plants, though, because they are the most common, all right? We can go outside and see plants pretty much everywhere, all right? So in order to understand how they photosynthesize, we need to know some very basic anatomy of the leaf, right? And like I said, I have cut this anatomy down to its very, very basic components here. So if I look at a leaf, right, and I cut that leaf in half, and I took a peek inside of the leaf, I would see something similar to this cartoon version up here. I see the top. This is the top of the leaf here that I would be able to see. This is the bottom of the leaf that I would be able to see. Everything in between, all right, we're talking microscopic level here, is really where photosynthesis is going to be concentrated. So these cells in between, we call them the mesophyll cells, all right? Just the name that they have, all right? These are the cells that will undergo most of photosynthesis for the plant. Right, so all of these cells in between. Right? If we look at our leaf anatomy, right, we know in photosynthesis so far what we have learned, right? We're producing oxygen and we're using CO2. Well, those gases have to get to those cells somehow. And so these mesophyll cells need to use these gases, and these gases will enter our leaf through these openings called stomata. Right? So openings on the exterior of our leaves. They are very, very small, microscopic. Right? They will open and close to allow the gases in or out. Right? So again, basic leaf anatomy here. Now if we zoom in on one of those mesophyll cells, this is where we're going to find our organelle of chloroplasts. Right? And this is the organelle where photosynthesis takes place. Right? And so our chloroplast, we'll go into a bit more anatomy on the next slide here, has a whole bunch of membranes in it here. But inside of those membranes, we have pigments that are embedded that we call chlorophyll. Right? And it's a chlorophyll pigment inside of our chloroplasts that are inside of our mesophyll cells, and that is what captures our light energy. All right, so let's zoom in on our chloroplast anatomy here because this is where the site of photosynthesis occurs. Chloroplasts have a lot of membranes, right? And this is really key to making this process work. So first of all, they have these two outer membranes, right? We have a membrane on the very outside. We call this the outer membrane. We have this a membrane towards the inside of our chloroplast. We call it the inner membrane. And that space in between would be the intermembrane space. All right? What the membranes we're most concerned about for photosynthesis are these membranes in these thylakoids. Right? So these thylakoids, you can think of them as simply sacs of membranes. Right? And there's a lot of them here. So a single thylakoid would be one of these little discs here. Right? If you cut those discs in half, you'd see that it's surrounded by membrane and the inner space is pretty much empty. Right? If we take these thylakoids and put them in a stack, right, we would call this a granum. Right? And then outside of these thylakoids, all of this space would be the stroma. Right? So the thylakoids, this is where photosynthesis is happening. Right? The stroma is the space outside of the thylakoids. And inside of our thylakoids, we call that the lumen. And you might see those words appear several times on the next few slides. All right, we're starting fast and furious today. So let's take a step back. I want you just to label this chloroplast anatomy. We'll take a couple minutes to do so. Also a great couple of minutes to clarify anything you might be stuck on. Yes, and ignore 6. We did not talk about 6 because it's not important for our discussion. quiet class today. Must be the weather. Weather's making me feel really good. All right, let's go through this anatomy. Let's start with number 3 here. What would number 3 be? Outer membrane. Yes, what would number 2 be? Intermembrane space, very good. What about number 1? The inner membrane. Let's look at number 5 here. What is number 5? A thylakoid, and a stack of thylakoids here, that would be a granum. What about number 4? Stroma, yes, very good. All right, basic chloroplast anatomy. We did not go over the lamella because it's just not pertinent to our information, and I don't want to give you information you don't need to know when there's already so much. All right, so now that we know kind of some of the anatomy, let's get into photosynthesis itself. All right, so photosynthesis, much like cellular respiration, is broken down into a couple of different stages. Now, cellular respiration has 4 stages, right? Photosynthesis only has 2, right? And in photosynthesis, we break that up into the light reactions, which are happening in our thylakoids here, right? The light reactions, we're using light energy. We are going to produce ATP, NADPH, and some oxygen. The other stage in photosynthesis, which we'll talk about on Monday, is the Calvin cycle. Right? The Calvin cycle is going to use the ATP and NADPH from the light reactions in order to produce carbohydrates, and most likely those carbohydrates are going to be glucose molecules. All right? So if we're starting with the light reactions, we have to understand light and its basic properties. So light is a type of electromagnetic radiation, right? And if we look at the entire electromagnetic radiation spectrum here, we see that there are multiple types of light, right? We have everything from gamma rays and X-rays to radar and TV and FM and AM radio waves, right? The portion of this spectrum that we can actually see ourselves would be this visible spectrum. And it's this very, very small spectrum in the entire range of electromagnetic radiation, right? Now, light is going to travel as a wave. And these waves can be short, they can be very long. And so if we look at our spectrum here, we see that they can be very, very short. Gamma rays are very short waves. They can also be very, very long. Radio waves tend to be pretty long. Right? The visible light overall tends to be in the middle, right? But our blue light tends to be on the shorter side of our wavelengths, and our red light tends to be a little bit longer of wavelengths. Right? So they travel as waves, but they also behave as a particle, right? And these particles are called photons. So don't get that mixed up with protons, like the atomic structure, right? These are photons. So you can think of it like photo, right? An image. So image, you need light to see the image, right? So we have our photons, and these are like discrete packages, right? Discrete packages of light, right? Now these photons, traveling as waves, carry different amounts of energy. Our shorter wavelengths have higher energy than our longer wavelengths, right? We will see this come into play in a few slides here, right? But for now, we have light. It's traveling as a wavelength, right? And it also acts like a discrete packet called a photon. All right, so these photons are what is basically carrying the energy that these chloroplasts will obtain. So if we have our photon, these discrete packages of light, when they hit an object, they can do 3 things, right? They can be absorbed and used, like in photosynthesis. We'll talk about that. They can be transmitted, or simply go through something, right? So we have our light, our photons are coming in, it's going straight through our chloroplast, it's not being used. Or they can be reflected, right? So they're coming in, they're hitting an object, they're being reflected back, right? Now in our chloroplasts, we have pigments, right? Pigments are molecules that can actually absorb these photons and use that energy. So we're not using the energy in these photons that are being transmitted through. We're not using the energy in the photons that are being reflected, but those photons that are being absorbed, right? This is where we're getting our energy. And it's these pigments that can absorb this energy. So pigments, they are molecules that can absorb certain wavelengths of light. And so in photosynthesis, a major pigment would be chlorophyll, right? And this is an image of our chlorophyll molecule here. Chlorophyll actually absorbs red, blue, and violet light, right? We're using those wavelengths. And it's reflecting back the green wavelengths. We're not using the green spectrum. And so that green light is reflected back. All the others are absorbed. And that's why when you look at a leaf is green, right? Because it's that wavelength being reflected back at us. Yes? Very slightly so, yes. So in our chlorophylls here, there's actually two main types of chlorophylls, chlorophyll A and chlorophyll B. They differ, and basically in oxygen and some hydrogens at this area of our pigment, right, they act in pretty much the same way. The wavelengths that they take in are just ever so slightly different, right? Not different enough that we're going to differentiate it in Bio 101. All right, we have our pigments. They are capturing light energy from these photons that are being absorbed, right? When these photons are absorbed, they are transferring energy, and they are doing this by exciting electrons, right? So we have, an electron orbiting a nucleus here. A photon comes in, strikes this electron, transfers energy to that electron. That electron becomes excited, right? It kind of moves slightly further away from our nucleus, right? It is now excited, it now contains energy, right? Now having an excited electron with all of this energy is really, really unstable for the atom. And so what the atom is going to want to do is get rid of that excitement or that excited state or that electron as soon as it can, right? So to get rid of the energy in this excited electron, this electron can do three things, right? It can release the energy as heat, right? So if you're ever driving on the road on a hot day, that black top, right, is releasing a lot of that light energy in the form of heat, right? Stepping on that black top on a really hot day might hurt because of all that heat coming off. The electron can release its energy in the form of light, right? So if you're familiar with animals that fluoresce, we call these bioluminescent animals, they are releasing some of that energy in the form of light. Right? But these excited electrons, they can also just simply be transferred to another molecule. And that is what is happening in photosynthesis, right? We are transferring that electron to an entirely different molecule. All right, so we have this photon. It's striking our chlorophyll head here. The head is this rounded ring structure here. The tail is what's actually embedded in the membrane of our thylakoids. And this electron becomes excited. Now remember I said light, depending on its wavelength, can carry different amounts of energy, right? So no matter the light, right, as long as the pigment can absorb it, it's going to excite the electron. But if we look specifically at the color of light or the wavelength associated with light, it can excite it to a different amount. So blue light has a shorter wavelength, contains more energy, and when it excites an electron, it excites that electron to an even higher energy state. Versus red light has a longer wavelength, doesn't have as much energy. It's still going to excite our electron, but it's not going to have as much energy associated with it. All right, so we can absorb red and blue light, right? Blue light's going to have more energy than that red light simply because of its wavelength, all right? Now we have our chloroplasts. We know that they're absorbing these photons, but they are not acting alone, all right? These are, excuse me, I said chloroplasts. Chlorophyll, our pigment chlorophyll, they are not acting alone. They will actually group into these complexes of like 200 to 300 chlorophyll molecules in a complex. And we call that an antenna complex, right? So they're not working by themselves. They're working with an entire group of chlorophyll pigments. And when an electron gets excited in one chlorophyll pigment, right, that pigment might hand off that electron to another chlorophyll, which might hand it off into another, and bounce back and forth between these pigments until it reaches what's called the reaction center in the middle of these groups of pigments, right? The process of moving that electron from chlorophyll to chlorophyll to chlorophyll to chlorophyll is called resonance energy transfer. Right? We're transferring our energy in our electron by simply bouncing it between all of these different pigments. All right, chlorophyll is not the only pigment that we find in plants, right? And that is especially prominent at this time of year with the leaves changing colors, right? So there are many other pigments in plants. These pigments can absorb different ranges of wavelengths. So chlorophyll, we know, will absorb red and blue and reflect back some green because it does not use green. Other pigments, like carotenoids or xanthophylls here, they can reflect in different or they can absorb different wavelengths, and they will reflect back as like a red or a yellow or an orange, right? In the fall, our trees out there, they're actually breaking down their chlorophyll pigments and moving those parts back into the trunk of the tree to be recycled. And what's left are all these other accessory pigments, which reflect different wavelengths of light, which means we are seeing our trees turn red and orange and yellows, right? We're seeing a lot of these accessory pigments right now, right? So they're absorbing in these different wavelengths here, right? Wavelengths that chlorophyll cannot absorb. And they can transfer that energy that they're absorbing to a chlorophyll molecule through that resonance energy transfer and to actually use those other wavelengths. They can use some of that green light, right? But they can actually act as a form of protection as well, right? So some of these pigments, they kind of act like the sunscreen of a plant, right? Light is another good example of too much of a good thing can actually harm a plant. All right, that was a lot of information, and we still have more to go here. But I want to just take a step back and review our pigments, all right? Answer these 3 questions here, and then we'll come back together. Yeah. All right, no one's talking. I'm going to take that as a sign that you have all answered these questions. So, who wants to tell me what is a pigment? Yeah. Yeah. They're molecules that absorb certain wavelengths of light. Yeah. What are 3 ways that an electron can release the captured light energy? Mm-hmm. So heat and light. Yeah, they can simply transfer that electron to another molecule. Yeah, make it somebody else's problem, essentially. Yeah.
So is heat and light like reflection?
Not necessarily. So reflection would be like reflecting light that it's not absorbing at all. But in this case, we're absorbing that energy, and then we're releasing the energy as heat or light. Yeah. All right. And then my final question here: does red or blue light excite an electron more? Blue, yeah. Why does blue excite an electron more? Has a shorter wavelength. Very nice. All right. Any questions before we move on? All right, we're going to get into the nitty-gritty of light reactions. And if we don't get through all the slides, that's okay. We'll just continue this on Monday here. So we're going to talk about the light reactions today. We'll talk about the Kelvin cycle, or the dark reactions, on Monday. So light reactions, we're in those thylakoid membranes here. We are using light energy and water to create oxygen, NADPH, and ATP. Right? NADPH is really, really similar to NADH from cellular respiration. Right? It's just ever so slightly a different molecule, but it acts in the exact same way. Right? So if you're a fan of Lord of the Rings, I saw this meme a couple years ago and I thought, oh, this is perfect. Right? So we have Sam as our NAD plus molecule, right? And Frodo as our electrons, right? And he can't carry the energy, but he can carry Frodo. He can carry those electrons. So when he carries those electrons, they become NADH. All right? Thought that was a good meme. It kind of helped me understand it maybe a little bit. All right, so we are creating NADPH, we're creating ATP, and we are creating some oxygen. And this is all happening in the membranes of our thylakoids. And so this is a great synopsis image here, right? But it does look overwhelming at first. Right? We will walk through this, and a lot of these processes are very similar to cellular respiration. For example, we'll see the electron transport chain again. We will see ATP synthase. All right. Importantly here, again, we are watching the movement of an electron. Right? Just like an electron moved through the electron transport chain in cellular respiration, an electron is moving through the electron transport chain and other proteins through the light reactions. All right, so our light reactions, they contain these very important protein complexes that we call photosystems, right? These photosystems, yeah, I'm tripping over my words today. These photosystems, right, are the proteins that will capture the light. They are the proteins that have the chlorophyll, right? Now there's 2 types of photosystems, right? Photosystem 1, which was discovered first, hence photosystem 1, And then photosystem 2, which is actually the first step in photosynthesis, right? So when we go through our light reactions, we'll go photosystem 2 and then photosystem 1, right? I don't know why they didn't change the name after they discovered the second one, but they didn't, right? So keep that in mind when we're talking about our photosystems. Photosystem 2 will come first. In both of these photosystems, again, we are exciting these pigment molecules. So if we look at the structure of these photosystems, right, we have that antenna complex, right? Those groupings of 200 to 300 of chlorophyll pigments. It's that antenna complex that will absorb the photons, right, take in that energy. And it's in these antenna complexes that excited electron will bounce between different pigments. Right? Now in the center of our photosystems, we have what's called the reaction center. So after that electron is done bouncing between pigments, it's going to end up in this reaction center. And in this reaction center, that electron is going to be quickly given away to yet another molecule. Right? But it's that molecule that will take it away from our photosystems and into the electron transport chain. Now our photosystems are a little bit different in the reaction center. Right? The reaction system for photosystem 2, which comes first, right, is called P680. And for reaction center, or for photosystem 1, it's called P700. Right? The numbers are a reference to the specific wavelengths that they can utilize in the end. Right? And I'm not going to get into the specifics of that because I don't want you to get bogged down with those details. But these are the two differences between our reaction centers. When they have an excited electron, we put a little asterisk behind it to say that the reaction center has that excited electron, and it is in this excited, unstable state. Right? And so because when it's in this very excited, unstable state, it wants to quickly get rid of those electrons, and it will be moved to yet another molecule. All right, so we have our antenna complex here. We have our light exciting our electron. That electron is bouncing between different chlorophyll molecules until it ends up in the reaction center in the middle. And that's this dark green bit on this image. When it ends up on a reaction center, that electron is going to be quickly given away to a primary electron acceptor, and then it will go through an electron transport chain. All right, so let's focus in on photosystem 2. Like I said, this is the first step in photosynthesis, even though it's photosystem 2. Okay. We are exciting that electron, and then we're going to pass it off to an electron transport chain. Okay. Now, photosystem 2 actually has this really unique property. It's the only known protein that's able to oxidize water, right, in this particular way. So we have this excited electron that's eventually going to leave the photosystem. We need to replace that electron, right? And that replacement electron comes from water. We are splitting water apart, right? When we split water, we're donating an electron, right? But we're also creating oxygen and some of these hydrogen ions, also known as protons, right? Because hydrogen ion doesn't have an electron. It just has one proton and a neutron, right? And I know I've gone over this so many times that I really want you to understand it here. So this is yet another image showing the exact same thing here. We have light coming into our antenna complexes, right? It's exciting an electron. That electron is bouncing between different chlorophyll molecules until it ends up at the reaction center. And for photosystem 2, this is P680, right? That is really unstable, so it's going to quickly give away that electron to a primary electron acceptor. But it needs to replace that missing electron, so it's going to split water apart to replace that electron, but it also produces oxygen, right? So we have this particular step in photosynthesis to thank for us being able to breathe in oxygen, right? I kind of already went over this slide here. This slide is just reiterating that we're splitting apart water, right? And that's giving us oxygen, that's giving us hydrogen ions, and it's replacing that lost electron in our photosystem too. All right, so that electron is now in an electron acceptor, right? A molecule that will take it away from photosystem 2 and then bring that electron through the electron transport chain, right? This part of photosynthesis works really similar to the electron transport chain of cellular respiration, right? The electron is traveling through different proteins, right, being handed off, and as it's being moved through these proteins, it's drawing in some of these hydrogen ions, right? We're going to eventually build up a large gradient of hydrogen ions here, much like we did in cellular respiration, and that will eventually help in making ATP, right? We're not going to go through all those specific proteins in the electron transport chain, but know that electrons are moving, hydrogens are coming into the thylakoid. All right, after the electron transport chain there, that electron's going to end up in photosystem 1, right? And so by the time it makes it to photosystem 1, that electron has lost a lot of that energy that it initially gained from the sun. We have to re-energize this electron. And so in photosystem 1, same thing happens, right? A photon is going to hit our photosystem. That's going to re-excite that very same electron that was excited in photosystem 2. And then that same electron will go again, once again, to an electron transport chain, right? So we have 2 electron transport chains here. All right. This is just another way of showing the electron transport chain. We have our photosystem 1 here, right? Our photons are coming in, they're exciting our electron. The electron is going through this electron transport chain, right? And the end result here is that that electron will be combined with NADP plus to make NADPH, right? So that electron is done with its journey. It has ended up in NADPH. That NADPH will eventually go on to the dark reactions that we'll talk about on Monday. So to put it all together here, I like to think of it as the Z scheme. So on our vertical axis, right, we have basically the amount of energy, right? If we start with photosystem 2, with our initial electron coming from water, we're exciting that electron and providing a lot of energy to that electron. That electron is then going to travel through the first electron transport chain, right, until it hits photosystem 1. By the time it hits photosystem 1, that amount of traveling that it has done has lost a lot of energy. We need to re-excite it. We are re-exciting it using light, right, to an even higher state of energy. It's once again going to go through an electron transport chain where it's going to lose some of its energy, and the electron will end up in NADPH. All right. Any questions about the movement of an electron? Yes. No, so the Z scheme is basically just all the reactions in the light reactions. It's called the Z scheme because if you turn it on its side, it kind of looks like a Z, right? Kind of like a zigzag. So it's not losing any energy because it hasn't yet been energized. Right? It gets energized at the beginning of this and then ends up in NADPH. Does that make sense? So the Z scheme is the light reactions. Any other questions? Yes?
What are the enzymes?
They're proteins. Yeah, we call it protein complex because there's lots of things. Yes. Yes. Not quite like an ATP synthesis. Yes. Yeah, they're embedded in the membrane. They're being excited, they're bouncing between the pigments embedded in the photosystem protein complex, and then eventually we'll leave it to go through that electron transfer. That clear things up? Okay. So it doesn't quite go through like ATP synthase. When it moves through ATP synthase, it actually provides a spinning motion for ATP synthase. But in this case, this protein complex is just housing the electron until it can be passed off into the electron transport chain. Mm-hmm. Yeah, good question. So in photosystem 2, right, there is really no product. I know on this one it says it's producing ATP, right? Indirectly, yes, it is producing ATP, and we'll talk about that in the next slide here, but there's really no product, right? The only product being produced is at the end of that second electron transport chain at NADPH. Yeah, yep, only producing one NADPH. But this is happening so fast in so many different proteins, in so many different thylakoid membranes, in so many different cells, that we get a lot of NADPH built up. Yes, good question. So we're talking about right here? Yeah, adding a plus and adding in a plus. Yeah, it's because we're adding in that negative electron. And in fact, it's not just one electron, it's actually 2 electrons doing this at the same time. So those 2 negative electrons will cancel out the positive charges. Yeah, good question. All right, so this is almost my final slide. My final slide of a lot of content here, right? This whole process that has been happening in our thylakoid membranes has been building up a proton gradient, or a hydrogen ion gradient, right? And because we have this gradient built up, right, that gradient will power ATP synthase, same exact way that it powered ATP synthase in cellular respiration, right? So in the light reactions, this gradient is built up in 3 ways, right? The first is when we split apart water to replace that electron, right? We're creating oxygen, but we're also creating some hydrogen ions, right? So that's populating these hydrogen ions inside of our thylakoid. The second is that electron transport chain, the first one, right? That movement of the electrons is bringing our protons across into the inner space of the thylakoids. Okay? And the last way that this gradient is being made is not by adding more hydrogens here, but decreasing the amount of hydrogens on the outside of the membrane. Hopefully my math doesn't die here. All right? But when we create NADPH, we're combining it with some of these hydrogen ions. We're using up some of these hydrogen ions. So there's even fewer outside of our membrane here. So that once again contributes to that gradient of having a lot on the inside and very little on the outside. Right? And then that gradient, right, those hydrogen ions will be pumped out through the ATP synthase, and we create quite a bit of ATP. All right, so light reactions, big picture. We are creating 3 things. We are creating oxygen, which is a byproduct, but we are very thankful for that byproduct. We are creating NADPH, and we are creating ATP. NADPH and ATP will then go on to be used in the dark reactions or in the Kelvin cycle. And that is much less complicated than the light reactions, you'll be happy to know. All right, any other questions I can answer? All right, so homework from Monday. Yes, there is another content check. I tried to keep it pretty basic because we just had a content check. Okay. I did schedule us an extra study session. This is not required, this is optional. Right? But I will be in this room next Wednesday from 4 to 5 just to answer any questions you might have. I won't have any content. I won't be passing anything out. I'll just be here to answer your questions and study with you. right? Do have a scientific literacy assignment due next Wednesday, and then of course the exam on Friday. Yes,
When's the final video?
on Monday. Yes, and it's much less content heavy than this lecture is. There's also a lot of additional resources, right? I have been told in particular, this interactive video here and this interactive game is super helpful in understanding photosynthesis.